Hello again. We will resume explaining the structure of the hair, but in more details. So I'm going to move on to the diagram, to the next diagram of the hair. Here we can see a section in the epidermis, a section in the dermis, and in the hypodermis. And of course, what are the structures we can see here other than the epidermis, the dermis, and the hypodermis, which are actually the layers of the skin? We can see the hair, and the hair is protected inside the hair follicle. We can also see a gland that is associated with the hair follicle, and we can see a slender muscle. And I say a slender muscle because you are supposed to draw it in a slender way on the diagram, which is called the erector pili muscle. So if I ever ask you about a muscle associated with the hair follicle, you say the erector pili muscle. What's the function of the erector pili muscle? As its name indicates, the erector pili muscle erects the hair or causes erection of the hair. What is erection? This means that the hair would stand perpendicular to the surface of the skin. What's the importance of this or the significance of this for animals, for example? It helps animals to heat themselves or to keep the heat of their body inside, because when all the hairs of the skin or of the fur on the skin are erected, if it's an animal, then these erected hairs would trap air in between them, and therefore this air would by conduction receive the heat of the body, and the hairs would keep the warmth inside the air in between. So this helps the animal keep warm or stay warm. The second importance of the erector pili muscle is that when the hair is erected, it appears of a bigger size, or the animal appears of a bigger volume, and therefore the animal becomes more scary to predators and to other animals that want to harm it. So this is another importance. In human beings, it is erection of the hair of the skin is mainly indicates that the individual is scared, or that the individual is in a very cold weather and he feels very cold, because the sympathetic nerves, this section of the autonomic nervous system, acts during cold weather to erect the hair to keep the human being warm. But actually, since we don't have much hair on the skin, these goosebumps are an indication that we are scared, or that we have strong emotions, or that we feel cold. The gland that is associated with the hair follicle is the sebaceous gland, and the sebaceous gland, as its name indicates, is a gland that secretes sebum, and sebum is a fatty material, and this sebum is poured into or secreted into the hair follicle, and this helps eruption, not erection, eruption of the hair, meaning that the young hairs can erupt or can find their way outside the skin, and afterwards it helps the hairs to be soft and pliable. So this is the importance of sebum. Is the sebaceous gland endocrine or exocrine? It is exocrine, since it has a duct that opens into the body cavity. Even if the duct is short, yet it is present. It doesn't pour sebum into blood, so it cannot be an endocrine gland. And meanwhile, we can see that it pours its secretion into a cavity. What is this cavity? It is the hair follicle, and afterwards sebum will be secreted on the surface of the skin. Just like we talked about the importance of the sebaceous gland, we must mention the potential harms that might result from the sebaceous gland in case it gets infected. When is it possible? the likelihood of infection of the sebaceous gland occurs at puberty when androgen and male hormone secretion increases. This affects the amount and texture of sebum, or the thickness and stickiness of sebum. So the gland starts secreting more sebum, and that those increased amounts of sebum are more sticky, they are thicker, and therefore there is plugging of the duct of the sebaceous gland and stagnation of the secretion. So the gland continues secretion of sebum, but sebum is not released from the gland. So the gland gradually increases in size and swells to the extent that swelling in the dermis might cause a bump on the surface of the skin, which we call acne vulgaris. And if the gland, which is very probable because of the blockage that occurred, if the gland gets infected, then those swellings will show signs of inflammation, which are again more swelling, redness, pain or induration, tenderness, and warmth. And this is what we call acne vulgaris. In addition to the changes that occur to sebum, we have changes that occur in the epithelial cells that surround the gland. There is more proliferation of those glands, and then the proliferated cells will die, and the dead cells will further share in plugging and obstruction of the duct of the gland, and therefore would increase the symptoms. What is the hair structure? Now, let's start by the structure of the hair follicle. As you can see, the hair follicle is nothing but an extension of the layers of the epidermis of the skin. So if I ever ask you about epidermis-derived structures, then the hair follicle is a good example for epidermis-derived structures. Why? Because as you can see, the stratum germinativum is continuous with the stratum germinativum of the gland. And all those layers in between the stratum germinativum and the stratum corneum, namely the stratum spinosum, the stratum granulosum, will be continuous until we reach here. And they would continue with a very thin layer above the stratum germinativum, and the stratum corneum is nothing but the dead epithelial cells that will be seen here inside the cavity of the sebaceous gland, and that would help in plugging of the sebaceous gland later on. So they are actually the same layers as the layers of the epidermis of the skin. They extend around the sebaceous gland, but they are thinner, and the cornified layers form the dead cells, which will obstruct the gland if they increase in number. When we come to the structure of the hair follicle, it's exactly the same thing. Stratum germinativum of the epidermis is continuous with the stratum germinativum of the hair follicle. Stratum spinosum and granulosum will be continuous with those of the hair follicle. And then finally, the stratum corneum. Where has the stratum corneum gone here? The stratum corneum is nothing but dead cells. So here, actually, we have those dead grayish cells in the inner root sheath. So the layers of the hair follicle from outside inwards are very easy. I might ask you, of course, from outside inwards or from inside outwards. If I ask you from outside inwards, then you would say, they are very easy to deduce, by the way, if you know the layers of the epidermis. So number one, most outer, you will find the stratum germinativum. And then afterwards, you will find the outer root sheath. So you have the stratum germinativum, it's like a separate layer, and then outer and inner. The outer root sheath is nothing but a continuation or continuity of the stratum spinosum and stratum granulosum. See this pink color? And then extended downwards, this is the outer root sheath. The inner root sheath is nothing but the extension and accumulation of the stratum corneum. But if we come to those three layers of the wall of the hair follicle, in their lowermost part, we will find that there is like an invagination, just like something pushed it upwards, invaginated itself. There is an inward elevation of the hair follicle, but still with the same layers. But this is the first difference. So there is an inwards or an upwards actually invagination caused by the dermal papilla. So what is the dermal papilla? The dermal papilla is part of the connective tissue dermis that contains blood vessels, arterioles, venules, and capillaries in between them. And it is the part of the dermis that provides the hair bulb or the lowermost part of the hair with nutrition and oxygen and drives away waste substances. So this is the importance of the dermal papilla. The germinal matrix above the dermal papilla is very active, of course, to form hair cells and to form the hair cells of the lowermost part of the hair, which is the hair bulb. The hair bulb is an onion-like structure just above the lowermost part of the germinal matrix, whose living cells are formed by the germinal matrix. It's the same way the germinal matrix forms the layers of the skin, and the same way the germinal matrix forms the substance of the nail body. Of course, the lowermost part of the hair, which is the hair bulb, is alive because it's very close to the germinal matrix and very close to the blood supply in the dermal papilla. But in that invagination, we can see melanocytes. And those melanocytes are very important because they synthesize melanin, and then those melanin granules will diffuse to the neighborhood, which are the cells of the germinal matrix, and then from the cells of the germinal matrix to the keratinocytes or hair cells that are present in the lowermost part of the hair. And this is an answer to the question, how is the hair, or how does the hair acquire its pigmentation? So the hair gets pigmented. How does the hair get pigmented? From the melanin that diffuses from the melanocytes to the germinal matrix and then to the keratinocytes of the hair bulb. The uppermost part of the hair, which is the hair shaft, that has most of it above the surface of the skin. But of course, as you can see, the hair shaft does not start exactly at the surface of the skin. It starts a little bit downward. The hair shaft is formed of completely keratinized and dead cells. And that's why when you get this question like, justify cutting a hair through the hair shaft does not cause any pain, because the hair shaft actually consists of dead keratinized cells and does not receive any innervation, any sensory innervation, and therefore it does not feel pain. Of course, if you detach the hair from the hair bulb, you feel pain, because when you detach the hair from the scalp, you're actually separating the hair bulb from the dermal papilla. So you're actually rupturing nerves, sorry, or injuring sensory nerves, and that's why you feel severe pain. In between, we have the hair root. As we said at the beginning of the previous video, that the hair root is the junction between the hair shaft above and the hair bulb below, and actually the hair root is midway in nature between the hair bulb and the hair shaft, meaning that it's not totally dead, it's not totally alive. Signs of keratinization start to appear. That's why we say that it is almost completely keratinized, because some cells are still alive. The layers we talked about, the germinal matrix, the outer root sheath, and the inner root sheath, are the layers of the hair follicle, but there are other layers for the hair itself. So be careful whether I am asking you about the layers of the hair follicle or the layers of the hair. If I am asking you about the layers of the hair itself, actually I'm asking about the layers of the hair root, because this is where we can see those layers. So we have an outer cuticle, and then a cortex, and then an innermost medulla. The outer cortex is almost made of non-nucleated cells, clear cells, and those cells, the outer, cuticle is clear, transparent, and it reveals the color of the cortex of the hair underneath. The cortex, which is the thickness between the cuticle outwards, the cuticle is protective, by the way. So the cortex in between, the middle layer of the hair between the cuticle outwards and the medulla inwards, consists of cells that contain keratin and melanin. Of course, as you know, keratin makes the hair waterproof and tough, whereas melanin gives pigmentation. And finally, we have the inner medulla, and actually the function of the medulla is not yet revealed completely. It's not very well known up till now. Concerning the hair color, of course, it's important to know that we have different shades of colors, and that the inheritance of hair color is a cumulative type of inheritance, meaning that the more you have of the dominant alleles that give you the black color, the more, or the dark brown color, the more your or the darker your hair is, and vice versa. So this is what cumulative inheritance means, and meanwhile, this is the first factor that determines the hair color. But there are other factors that determine hair color. For example, exposure to the sun, exposure to chemicals in swimming pools. You know that some chemicals in swimming pools decolorize the hair and make it more or less, I can't say blonde, but more or less yellow. So exposure to chemicals affects the hair color. Exposure to the sun affects hair color. Genes affect hair color. And okay, and that's it. Of course, the use of hair dyes, for example. Aging affects hair color, because you know that as we go older, we get older, we will have fewer melanocytes in the germinal matrix of the hair bulb, and that's why the hair gets grayish or whiter. And now we're going to talk about the diseases that are related to the hair. As I told you last time, you will have a clinical case study. For example, if you have a case of Taenia capitis or ringworm infection, and I would tell you that a patient comes to the clinic complaining of a localized hair loss, explain how through the history, through the examination, and through the investigations, you can diagnose the disease. So first of all, the personal history, of course, the name of the patient, the age of the patient, the marital state of the patient, where the patient lives, and the patient's job. All these are important because they might have a relationship to the present disease. And then, for example, if the patient is divorced, then this might be a stressful condition that elicited, for example, alopecia areata, or it would help in differentiation, because you know that localized hair loss may be Taenia capitis or may be alopecia areata. Then we come to the present history. And in the present history, we start by analysis of the present complaint. Again, in the present history, we start by analysis of the present complaint. We start by analysis of the present complaint. What does it mean, starting by analysis of the present or main complaint? Meaning that we ask ourselves what drove the patient, or what triggered the patient to come to the clinic. It is the sudden appearance of a localized patch of loss of hair in the scalp. So this is the chief present complaint of the patient. So I will start by analyzing this chief main complaint. So I'll ask the patient questions, and then I'll report the answer. So I would say that the patient came to the clinic complaining of a localized patch of hair that was discovered a week ago. For example, this is the onset and the duration at the same time. It was suddenly discovered. This is the onset. It started a week ago. This is the duration. And it's progressive, meaning that it started as one patch and it ended up through over the week by showing 3 patches. So this way, I talked about the onset, I talked about the duration, and I talked about the course. Is it progressive or a regress? After having discussed the onset, the course, and the duration of the present complaint, I would talk about the site, which is again part of the analysis of the present complaint. So for example, I would say that the patient says, and I haven't yet examined the patient, so I cannot say in the present history that I discovered or I saw or I observed, because I haven't yet examined the patient. So I would say that the patient says that these patches, for example, are in the central part of the skull. These patches are in the central part of the skull, or in the anterior most part of the skull. Okay, so onset, duration, course, and now site. And then I would say that the patient says that the lesion is itchy and that he feels that the lesion is warm after having itched for a while, or after having after having felt itching for a while. The patient feels that the area is hot or okay. Then I would continue in the present history. I analyzed the present complaint, and I asked about associated symptoms like itchiness, like redness, like heat at the site. I asked about the location of the lesion, and then I would ask about any treatment received, and I would report this. And then after having finished the present history, I would ask about the past history, whether the patient had similar attacks before or not. And then I would ask about the medical history of the patient. Are you diabetic? Are you hypertensive? Do you have any immunological disorder? And then why diabetic? Because diabetes increases the incidence of fungal infection. Why hypertensive? Because this might be an indication of exposure of the patient to continuous stress. Why immunological disorders? Because alopecia areata, a related condition, is an immune disorder. So I ask about medical disorders, but medical disorders that might be associated with the condition. And then I end the present history by saying that upon asking the patient, he said that he does not have other complaints related to other symptoms. The family history is important. And then I would examine. Upon examination, and then I report the results of the examination and say, upon examination, 3 patches of localized loss of hair were observed in the central part or in the anterior part of the skull. They were red. They showed signs of inflammation. There were white flakes, and there was broken hair. So, of course, this leads you to examining under the microscope or under the dermatoscope, where you can see 2 things by the dermatoscope. You can see the ringworms in place, or of course, if it is alopecia areata, you wouldn't see them. And at the end of the day, you would see the broken hairs. Okay? We can do a skin biopsy, of course, but in such a case, it's not needed. So this is concerning Taenia capitis and alopecia areata as diseases. Concerning keratin hair treatment, I might ask you to justify the use of keratin. In keratin hair treatment, you would say that keratin is a tough protein. Keratin is a waterproof protein, and therefore keratin makes the hair look straighter. It would add gloss and thickness and shine to the hair, and it would reduce the frizz in the hair. Type, what are the harms? Keratin therapy involves heating, and therefore there are fumes that arise during the process. And these fumes contain a chemical added to keratin that is called formaldehyde. And the formaldehyde fumes are inhaled by both the patient or the person in question that has the keratin hair treatment done, and the hairdressers themselves. And therefore, this formaldehyde on the long run and on repeated use might cause irritation of the lungs and cancer lungs, even if the patient uses a mask. Of course, I wouldn't go over the glands, but I would go over the skin farming. What is skin farming? Skin farming is commercial growth of the hair, commercial growth of the skin, meaning that we prepare skin on commercial basis. What's the meaning of commercial basis? On a large scale. How do we use this skin or those skin pieces that are produced on a large scale? We use them to cover chronic wounds or to cover chronic burns that do not want to heal. Any chronic burn that doesn't want to heal, any chronic wound, that doesn't want to heal, or any chronic burn might be disfiguring and might be a source of infection. So we have two risks here. The first risk is the psychological status of the patient. The patient may feel depressed because of this disfiguring wound or burn, and at the same time the infection might spread because this is a dangerous area. What do we use in order to carry out skin farming? We use stem cells. What are stem cells? Stem cells are undifferentiated cells. When they are undifferentiated cells, this means that they are unspecialized cells. And why do we use stem cells? We use stem cells because, being unspecialized, they have the power to specialize to any other type of cells when they are put in a suitable culture medium. Of course, the older the cell, the less undifferentiated it is, the more specialized it is, and the less the power of the stem cell to give us any type of tissue. So if you use early embryonic stem cells from aborted early embryos, this is the best type of stem cells because they are totally undifferentiated and they are totipotent cells, meaning that toti is like two in French, which means all. They can differentiate into all types of cells. But if you use stem cells from the foreskin that is removed during the operations of circumcisions, then in such a case, they are multipotent cells. They can differentiate into multiple types of tissues, but not all types of tissues. The infant foreskin stem cells gives you a skin that is rich in keratinocytes and fibroblasts, but it might not contain sweat glands and sebaceous glands or melanocytes. So it might have a different a skin tan that is, or a skin shade that is different from that of the rest of the skin. And finally, epidermolysis bullosa. Of course, epidermolysis bullosa is, we forgot to say that if your previous condition of localized loss of hair was alopecia areata, you would find that there are family members that have autoimmune diseases, not necessarily alopecia areata, but any kind of autoimmune disease, scleroderma, systemic lupus erythematosus, and so on. So when you have a family member that has a certain autoimmune disease, you do not necessarily have all members having the same autoimmune disease, but you will find genetic predisposition and cases of different types of immune diseases. So it's worth mentioning that alopecia areata is an autoimmune disease, an immune disorder where the immune cells attack the hair follicles and destroy them. And that's why we can use cortisone and immunosuppressive drugs to suppress the overactive immune system and treat the patient. Another thing that's worth mentioning in alopecia areata, that it is related to stress, and therefore you might ask the patient about any triggering incident like separation of the parents or separation of the patient itself from their partners. This is important in such a case. In epidermolysis bullosa, this is a genetic disorder. And genetic is different from congenital. Genetic means that there is an abnormal gene, and that it is inheritable. It is transferable through offsprings. But congenital is not necessarily genetic. For example, if the mom doesn't have any abnormal genes, but she takes harmful medication during the first trimester, during the first 3 months of pregnancy, and the fetus or the baby is born with an abnormality, this is not a genetic disorder. This is a congenital disorder. Of course, needless to say that all genetic diseases are congenital, but not all congenital diseases are genetic. In epidermolysis bullosa, this is a genetic disorder. We have a gene, a recessive gene that is mutant. It is a mutant recessive gene that results in an abnormality in the proteins of the skin, namely keratin and collagen. The abnormality in keratin means the skin less tough and less resistant and less waterproof, so it absorbs water easily. The abnormality in collagen and elastin are very important in tightening the connection between the epidermis and the dermis. So you will find that because of this abnormality in collagen, the epidermis will slough off or will separate from the dermis because there isn't adequate collagen strength to tie them together, or to tie them together actually. And meanwhile, the skin itself is very weak and imbibes water easily because of lack of keratin. So you will find sloughing of the skin, sloughing off of the skin. You will find blistering, and a blister is a separated layer of the epidermis that is filled with fluid, and therefore patients are very sensitive to touch, to friction, and they easily get wounds and ulcers on the surface of the skin. So skin farming would be very important. Why is skin farming important in epidermolysis bullosa? Bullosa because there are bulli, or there are vesicles. Epidermolysis because there is lysis of the separating layers between the epidermis and the dermis. Skin farming is important in such cases to cover the ulcers, diminish the infection possibility, and diminish pain, because when the skin is sloughed off, then we would have pain because of exposure of the sensory pain nerve endings, and meanwhile, because of the absence of the mechanical barrier of the skin, we would have increased exposure to infection. Thank you so much, guys, and I hope you would, inshallah, you would find the exam easy. I don't think I would have time to give you or to send you a video on the first part, which is the skin. But if you have any questions about it, please send them on WhatsApp and I'll be answering them. Thank you, guys, and see you, inshallah, tomorrow. And good luck with your test, inshallah. Bye-bye.